Arizona State University and Science are recognizing researchers who address pressing problems. The ASU Science Prize for Transformational Impact honors early career scientists developing innovative solutions to today's biggest challenges. Hear from the inaugural winners on ASU's Lab Coat Optional podcast and visit science.org/ASU to apply. This podcast is supported by the Icahn School of Medicine at Mount Sinai, an international leader in research, education, and patient care. The medical and graduate school is part of the Mount Sinai Health System, one of the largest academic medical systems in New York City. Ranked among the top recipients of NIH funding, researchers at Mount Sinai have made breakthrough discoveries advancing the health of patients. Here, clinicians and scientists push the boundaries in cardiology, cancer, immunology, neuroscience, genomics, geriatrics, environmental medicine, and artificial intelligence. The Icahn School of Medicine at Mount Sinai, we find a way. This is the Science Podcast for June 11th, 2026. I'm Sarah Crespi. First this week, new data suggests the Atlantic Ocean's circulation may withstand climate warming better than feared. This is with producer Kevin McLean and staff writer Paul Voosen. After that, we have researcher Scott Merrick. He talks about his study correlating socioeconomic conditions with brain structure and function in children.
Circulation patterns in the ocean move massive, almost incomprehensible amounts of water all over the globe. This circulation affects climate and carbon storage, making some places much more habitable than they otherwise might be. In order to study these patterns, you have to actually go out and measure them. And that's just what Paul Voosen did. Or I guess he wasn't doing it, but he hopped on a research vessel that was measuring the Atlantic Meridional Overturning Circulation, or AMOC, which scientists have been particularly concerned about in the last few decades. He's here to talk about the work they were doing and what's going on with the AMOC. Hi, Paul. Welcome back to the podcast.
Hello.
Tell me a little bit about your time on this research vessel. Have you ever been on a research trip like this before?
This was the first time I had the chance to do a full ocean-going research vessel. This was on the Royal Research Ship Discovery, one of the two flagships of the UK academic fleet. And they maintain this series of moorings out in the Atlantic Ocean along with US colleagues.
How long were you out there?
It's actually a very doable trip. It's a 8 or 9 day cruise, which typically when you think of research cruises, you're thinking like 2, 3, 4 weeks. So this is very humane.
Enough time to get a little bit seasick or no?
No, but we were also in the, maybe a few people did, but we stayed close to the Canary Islands. So off of Morocco, and we were kind of in their shield from most of the weather to the northwest. We were mostly to the southeast of them.
So you mentioned the moorings. So that was the purpose of this trip. They were going to check on all these moorings?
There's a series of moorings that stretch across the Atlantic Ocean. So if you think of the ocean like a big bowl of water with two sides, on one side, you have the US and Caribbean, and there's a series of moorings there that go from the ground up to the entirety of the water column, pretty much. And then on the eastern side, you have this series going up the kind of wall of the eastern side. You put these together, and that allows you to measure this circulation across the ocean.
What kinds of measurements are they taking?
Essentially, the basics of oceanography, which are salinity, temperature, pressure at different levels throughout the water column. When you add these all together, the ocean is stratified into these layers. You're able to kind of recreate pressure differentials on the ocean at the two sides of it, kind of like pressure differentials in the atmosphere that people use for forecasting weather. Comparing these, you can actually get a sense of which direction these flows are going in large scale.
It's so wild to think about giant air masses in the atmosphere, giant water masses in the ocean. In order to actually measure them, you have to take very simple measurements and then understand the patterns between them.
Simple measurements, but very complicated to take in that they have to be very carefully calibrated, immersed in the water, compared against ground truth, measured against water collected as you're calibrating it before they're put on the mooring, and then the same thing done to them when they come back.
Tell me a little bit about what was it like on the ship when they were doing all these collections?
It's very impressive. So the rapid array has been going since 2004, so 22 years. They're both collecting these moorings. So they get released from the bottom of the ocean floor with a acoustic signal sent to a clamp that releases it. And then the float's allowed to shoot back up in 5 to 25 minutes, depending on the depth. They collect it. They use grappling hooks and then pull it back to a hook and winch and take it back up. And then they do the same thing, putting them back in about the same place. And essentially, they assemble these moorings on the go as they're lowering it down, putting the sensors on. And then at the same time, they're always doing these CTD casts, these kind of metal frame that they put in the water to measure salinity of all those variables in the water column.
That sort of helps verify the measurements from the moorings as well.
Yeah, it's just what research ships love to do most of all. This is just, they're always doing CTDs. We're here, let's do a CTD. Let's do another CTD.
Yeah. It was interesting in your story to read about people that are waiting to get the first sight of what they're looking for in order to grapple it and everything. I remember being on a research ship years ago where folks were avid, I guess, spotters of all the equipment that they were looking for out in the water.
Yeah, it's always a little bit of a competition. It was an added competition because this cruise, I guess my interest in joining it a year ago kind of kicked off. They actually ended up bringing on a British film crew as well, or TV crew. And then the CEO of the National Oceanography Center in the UK also ended up going on it. So they had their own in-house photographers. So there's a lot of people on the bridge. So, a lot of competition, but the chief scientist, Ben Mote, was still able to spot it first.
Nice. Could you maybe walk through a little bit of the process of the AMOC flow? I know it is maybe a little bit easier looking at a diagram, which there is a great one in the story, but can you walk through what that process is a little bit?
In the Atlantic, you have this kind of, it's called the thermohaline circulation, right? This flow of water goes from south all the way up to the Arctic. It's pulled there by what's called the creation of deep water formation. So essentially, these waters, they get to subpolar or Arctic, they cool off, which makes them more dense. They form sea ice, which has brine rejection. They get saltier, which makes them heavier, and they sink down. And that sinking, you can think of it as like pulling the current along northward through the ocean. And it comes back down, flows back south, and connects with the broader overturning circulation that goes around the Southern Ocean, connects to all the other oceans. So it's not just about the Atlantic, but people really focus on the AMOC because of its power in providing warmth to Europe, especially, and other things.
Folks that have maybe taken an earth science or oceanography class may have heard of a conveyor belt, but it sounds like researchers don't love that term so much.
Yeah. It's a classic science communication thing, right? Wally Broker is a famed climate scientist, coined this idea, and it's a very powerful idea, and people still use it. It's this conveyor belt of heat, taking heat from the tropics up into the North Atlantic and giving Ireland and the UK and Norway a warm bath. It's much more complicated than that.
Right.
And, the best way to think of it is not one belt. It's a belt of belts that can all be going at different speeds or disconnecting from each other and then reconnecting. And, these interactions are much more complicated than the cartoon version traditionally presented.
What are some of the patterns that researchers have been seeing with the AMOC, and why have folks been concerned in the last couple decades?
There's been every reason to be concerned. In paleoclimate, so there are records of past climate change when we came out of the ice age, and there's this big meltwater events from the ice sheet that used to cover North America. Has been thought to be an evidence that this fresh water disrupted AMOC and shut it off, and shown that this can exist in like a bistable state. So it can be like turned off, turn on. And then climate models have also shown that this is a feature of the current. They haven't necessarily forecasted that it would happen, but they've shown that this is a thing that can happen. This has been true of climate models going back for a long time. So it's definitely a strong forecast that it would grow weaker. That's kind of been seen for decades. People thought just warming alone should prevent some of these denser waters and perhaps mute it. It hasn't been as clear from observations, because it's really hard to observe, that this is actually happening. And that's what these scientists have been out measuring for a couple of decades. And the evidence is still not totally clear.
Yeah, you mentioned a little bit in the story about maybe this cooling can happen further up and that could maybe buffer this process a little bit.
Yeah, so there's the classic view of deep water formation. So there's another array called OSNAP, which we'll see how much longer it lasts. It's primarily sponsored by the US. But they initially went into this thinking, oh, deep water formation happens in this Labrador Sea between Canada and Greenland. They found it's important in like the Iceland Basin, Erminger Seas, and then there's been other more recent evidence and work outside of OSNAP suggesting this deep water formation can happen up into the Norwegian seas, the Nordic seas, and then up into the Arctic Ocean that can migrate. And that could provide some buffer, some resilience to AMOC as warming continues. Now, geography is limited, right? So, this doesn't necessarily last forever. It's not a all clear thing, but it has been a factor that climate models haven't really well captured.
One of the things I wanted to ask you about was a new term for me, the unit of measurement for these water flows. How do you say that?
Sverdrup.
Sverdrup.
Well, I think that's right.
Yeah. What exactly is that?
This is just like great term for oceanography. So, I mean, essentially, easiest way to think of it is one Sverdrup is pretty much equal to the flow of water of all the world's rivers combined. When that's your unit of one, that tells you the difference between the ocean and rivers right there. The AMOC is generally thought to kind of move between 15 and 17 Sverdrups of water.
There's a meeting to sort of debate these AMOC changes. Are the same people that were on the ship going to be there?
They'll definitely be there. I think it was called AMOC collapse. Can it? Has it? Will it? From the press coverage, you would think that every oceanographer is kind of uniform in thinking that this risk of collapse is more likely than we thought before and near. And it's definitely not that widely agreed upon in the scientific community. And there's something to be concerned about, but this question of, is this something to really prioritize versus all the other impacts of climate change? And even in paleoclimate, the evidence may not be as clear as once thought that it did collapse. And climate modeling as well, these high resolution models, some of them seem to show it be a bit more resilient than thought, but not all of them. Will be some of the things I'm sure they're debating.
Thanks so much for joining us today. Great to hear about your trip.
Oh, yeah, my pleasure.
Paul Voosen is the Earth and Planetary Science reporter at Science. You can find a link to the story we discussed at science.org/podcasts.
No one goes to Hank's for his spreadsheets. They go for a darn good pizza. Lately, though, the shop's been quiet, so Hank decides to bring back the $1 slice. He asks Copilot in Microsoft Excel to look at his sales and costs and help him see if he can afford it. Copilot shows Hank where the money's going and which little extras make the dollar slice work. Now Hank has a line out the door. Hank makes the pizza, Copilot handles the spreadsheets. Learn more at m365copilot.com/work.
Choice Hotels get you more of what you value. Here's a little tune to help you remember. Same drive, different day. Don't you wish you were getting away? Pack your bags and come on through. Texas, Ohio, Alaska, we're up there too. Comfort Inn, it's calling your name. Save on the stay. Oh, and free waffles are yours to claim. Well, I hope you like my little song. Book direct at choicehotels.com.
On the site this week, we have some really great news stories I wish we could have covered. One by staff reporter Fee Jacobs about how so many theropod dinosaurs ended up with tiny arms. Also, a story on ancient DNA by Perry Thaler. This is on ancient DNA from squirrel poop or coprolites that lived 700,000 years ago. What can that ancient DNA tell us about the ancient world? And from Science Insider, our policy vertical, we have a story by the editor of that section, Jocelyn Kaiser, about the NIH plan to cap the number of grants a researcher can have at once. From the commentary section, we have our summer reading special section. This is a science fiction roundup. So there's a lot of really interesting stories on here. I'm just going to call out one that caught my attention. This is called Lake Effect by Hilary Berman, reviewed by Anna Farrow Henderson. Henderson writes that this book, Lake Effect, weighs the experience of caretaking for both givers and receivers in a collection of stories deeply engaged with the landscapes of the Pacific Northwest and the Great Lakes. I definitely want to be reading about cool lakes and gentle climbs during this hot, hot summer. All right, last up, I'll mention from the journals in Science Advances. There's a paper out by Manuel Pinheiro on genomic evidence for limited insect eating in ancient Europeans. So, what do the genomes of ancient Europeans tell us about their insect eating habits? Very cool. Okay, we're going to go on to the last segment of the show. It's on how childhood environments shape the brain with researcher Scott Merrick. Genome-wide association studies, this is GWAS. In those kinds of studies, you scan whole genomes and look for very small differences in the DNA between individuals. Then you apply statistical analysis to link those tiny changes with behavior or with disease. You don't know what the tiny changes means in terms of gene function, or even if those changes are in genes, it could be some other part of the genome. But GWAS are kind of a tool to narrow down where we should be looking genetically when we're trying to talk about something that's going on with a person or a disease. The science paper that we're going to talk about today by Scott Merrick and colleagues is about brain-wide association studies, BWAS instead of GWAS, which is a little bit different. As far as I can tell, you look at the structure of the brain and some of the ways it functions, and then you say, there are differences between people. Let's do math and see how those differences relate to environment, behavior, and disease. Scott Merrick, thanks so much for coming on the show.
Yeah, it's great to be here. I'm a huge fan of the podcast, so I'm happy to be able to take part in it.
The math is important, the statistics is important, but also maybe people don't really grasp how different brains are from each other. Like that was kind of something I went into this not really understanding. What is different between my brain and your brain?
That's a great place to start, really. How much of my brain function is similar to your brain function? And so we call it like group level variability. And it turns out that that's about half. So, the part of the brain, say, that controls your hand is in relatively the same location in everybody, right? Like it's not in the back of the head for you and in the front of the head for me.
I've seen the brain maps that say vision is here, hearing is here.
Right. And so the overall pattern is the same across everyone. However, there are individual specific differences from person to person, especially in higher order networks that subserve control and attention and things like that. There you can actually see pretty big differences person to person, especially networks that are located in the frontal lobes. So about half is shared and about half is individual specific from any one person to the other.
And just like with genetic studies, this requires a large sample size. Can you talk a little about how many brains you need to look at in order to say, oh, this is a reasonable difference that we can say is significant?
The real nuance here is the study design and what the expected effect size is given the study design. If you're doing this sort of observational study that we're doing here, that's the brain-wide association study that's similar to genetics. And there, similar to genetics, you need large samples. We don't need quite as large as genetics because the effect size is a little bit bigger. But, the effect sizes tend to be on the order of a correlation of, say, 0.1 to 0.2. And for that type of study, you need samples well into the hundreds or thousands.
If you've got a lesion on a brain, if you've got activity that, you standardize and you're looking at the brain as it's engaged in the activity, you don't need as much of a sample as you do if you're just looking at a huge sample and trying to pick out these like delicate changes person to person.
Right. And so I'm on studies where there was a sample size as small as 3 because some people casted their arm and asked, how does the brain change when you cast an arm for several weeks? And there, the effect size is quite large. And so you can see that in one person. You don't need 1,000.
I never even thought about how that would affect your brain.
Yeah, it's a really cool study.
Another broad question I guess I should ask here is when you're doing a brain-wide association study, you're looking at structure, you're looking at function, and you see differences between people. Those differences, are they meaningful? Like with the genetics, we could say it's a single nucleotide difference. Like, is it meaningful? We don't know. Can you say that in these kinds of studies?
The term meaningful is pretty loaded.
Yeah, good point.
I've always said it's just like a different goalpost, right? There's like the statistical significance goalpost, and then there's the meaningful goalpost.
So it's measurable, but what are we measuring, I guess?
Right. And so, here, like with the strongest association essentially being a child's census tract or, their zip code, using more powerful multivariate machine learning techniques, 16% of the variance between kids' brains could be accounted for by their zip code. I would say that that's a lot. That's very meaningful. But yeah, I suppose it depends on what lens you're coming into it with.
This is a really complicated study. So we start out with there are differences between brains. You need a big data set to find them. And then you can take those differences between brains, which are both structure and function, and say, what do they correlate with out there? And we're going to look at childhood environment, cognition, health, all these different things. And it's kind of a big pile of information that you have to kind of wind through. So let's just take first what you are measuring about these brains and what brains we're talking about.
The study is the ABCD study, which is the Adolescent Brain Cognitive Development Study. It's been going on since 2017 at 21 sites across the United States, which really helps in terms of representation, right? We're measuring kids' brains from West Coast, East Coast, middle of the country, et cetera. We're collecting a whole host of brain measures and a whole host of behavioral measures. So the brain measures that we're looking at here, one is cortical thickness, so how thick their gray matter is. And then the other is resting state functional connectivity. And it's a loaded term for how correlated each area of the brain is with every other area of the brain. So we take that information and then we correlate it with a whole host of variables that the ABCD study collected. So things related to socioeconomics, cognition, demographics, screen use, physical health, mental health, culture and environment. And yeah, it's essentially running a giant correlation study.
If I were taking that connectivity test, what would it be like?
You lay in the scanner and you stare at a crosshair, and that's it. So that's the really cool thing, is that we can tell a lot about individuals' brains at rest. Most of what makes us different, you can measure at rest. Most of what the brain is doing, you're not aware of. It's really cool.
And that's just like how in sync the different regions of my brain are.
Yeah. So the literal measure is how correlated, the Pearson correlation between a given region and every other region. And you do that for every region of the cortical surface. You end up with this big matrix of region to region, how correlated each region is with every other region.
And so if I were sleep deprived and stressed out, would I have lower correlations between some of my regions?
Different. So it would be altered in a very reproducible way.
How old were the kids in this study?
Ages here are 9 and 10. And then we ran some supplementary analyses when these same kids were 11 and 12.
And it was like a couple thousand?
Yeah, so the total sample size of ABCD is over 11,000. The ones that were included in here was 4,579.
What were you trying to figure out? Like you mentioned, you included behavior and environment. Like what were you trying to figure out from this big data set?
Essentially, how do kids' brains vary, or how do they differ? And what matters most for child brain organization, child brain development? Because we had so many variables to explore, we looked at everything that we could. And we just asked the question, what variable or what class of variables has the strongest association with brain structure and function? And it turned out that overwhelmingly that answer was variables related to a child's socioeconomics.
You did see this correlation between like socioeconomic status and these differences in kids' brains. This was a certain age group, so 10, 11-year-olds. Do we know how stable such an association would be?
So we do know, we looked in the UK Biobank data set, which is individuals 40 to 69. And also one of the largest associations in that data set is with a person's socioeconomics, and importantly, the brain pattern. So the regions of the brain that correlate most strongly with socioeconomics were very similar in that data set as in the ABCD data set. So there's a clue there that it might be more of a lifespan phenomenon, but that definitely is an area that needs to be explored more.
And what regions are we talking about? Where are you seeing these differences?
So the largest differences were in sensory and motor systems, which initially surprised us. We thought that it was going to be in attention and control systems, and that was not the case. And so we had to do some digging. This is where the brain patterns are really cool, because the brain patterns give you a clue as to the mechanism that's going on. You can't infer causality, but it certainly gives you a clue beyond correlation. That sensory motor map we found in looking at other imaging modalities, that pattern is highly similar to norepinephrine receptor density, sleep, stimulant effects, and other arousal related phenomenon. Another big surprise was that IQ also showed a very similar pattern to that. That was really surprising because IQ is purported to measure cognition, and regions that support cognition are not sensory and motor regions. They're in frontal and parietal cortex, right? If I put a person in the scanner have them do a working memory task or a control task. It's prefrontal cortex and parietal cortex that you have the strongest associations with. So that was a big surprise.
Was it because IQ and socioeconomic were linked?
So it does have to do with the fact that they're linked. So the correlation between SES and IQ, just the variables themselves in ABCD, is around 0.31. But the brain pattern was a lot stronger than that. So the brain map, the whole brain map, right, like the brain-wide map of IQ and the brain-wide map of socioeconomics, those were correlated at 0.72. It's very strong. So essentially overlapping neurobiology. And that's what gives us a clue that there's sort of this common neurobiological mechanism that's underlying those two things. And that's what we dug into more in the back half of the paper.
Kind of focusing on the fact that it's these specific regions that are different in these kids. What's a possible mechanism for that? Like, why would that happen? And what would it seem like in behavior?
The power of this data set is that it acts like a signpost. And the signpost to us, the thing that's screaming to us, is that this has to deal with sleep and stress. So kids that are living in poor neighborhoods are not getting as good of sleep and their brains are more stressed. It's not a brain that's innately worse at working memory or reading ability or anything like that. These are brains that are sleep deprived and stressed out. So for me, it's a signpost that for future studies, that's where we should be looking in terms of potential mechanism is for improving sleep and decreasing stress. The thing that needs to be conveyed is that SES, and in particular, a kid's census tract of their zip code, has the largest association with their brain. So a primary way in which kids' brains differ, or the primary way, is zip code. I think the sleep and stress pattern is the likely
Linkage.
linkage there.
I guess there's been a lot of debate about how useful IQ is for these kinds of studies. Is this research kind of showing that that's also true?
Yeah, that at the very least, we should probably be pouring more resources and attention into the socioeconomics and sort of the biological embedding of socioeconomics rather than trying to relate the brain to IQ.
I just wonder, like, where does it go? How do you kind of get traction on what's actually happening in people and how you could map this out, like the effect of sleep on brain, the effect of stress on brain, and then that it hangs around for a couple decades?
Yeah, and it's hard to know. That's one of the things we talk about towards the end of the paper is the extent to which the effects that we find are chronic or acute. Is it that the children are stressed and tired and so they're more drowsy in the scanner? Is it that they're just chronically more tired and they're chronically stressed and that's more of a chronic readout of their brain and it would take heavy intervention to lift them from that? When in development does this start? And then how pervasive is that throughout the lifespan? And then how predictive of later life outcomes is that pattern? All open questions. So that, my hope is that, yes, it is correlational, but that there are a lot of signposts here. And it as sort of like a guiding light for future research and funders to say, hey, we should be looking in these areas more, in particular, childhood sleep as a really big one, because it's clear that the kids are not getting enough sleep. The other thing I'll say, this is maybe a little bit in the weeds, but I'll try to communicate in a way that it's not. One of the things that MRI is most sensitive to in terms of brain function is a person's arousal state or their vigilance state. Essentially, like one of the main things that you can measure out is arousal. And then one of the main ways in which people differ is their zip code. And I think that a person's zip code is strongly loading onto their arousal state. And I think that's why we find that it's the largest association, is because you have this measure that's sensitive to arousal. You also have another measure that we're finding out is very sensitive to arousal, and that's a person's zip code, because sleep and stress are loading onto that. And probably the reason why IQ scores are as well is that, again, they're not actually measuring wholly the thing that we want them to be measuring. They're also picking up on this arousal related component. So that's why I think in terms of mechanism or where we should go, it's sort of barking up that arousal or vigilance tree. Like, I think that's where the explanation lies.
Are we just finding sleepy people? I mean, people who are chronically tired?
Could be. But that it's really interesting that like one of the main ways in which kids' brains differ is that. And it's going to have a lot of serious effects down the line, because that is related to their overall mental health, their overall cognitive status, which has rippling effects throughout the rest of their life. So yeah, it is. I think it's something that's very fundamental to what it is to being human, which is something that we all need, which is sleep.
And comfort and not being stressed out.
Yeah.
Absolutely.
Yeah, because I have 2 grants going in tomorrow. And let me tell you, when you are stressed out, it messes with your sleep then messes with your stress, and it's a vicious cycle.
And it definitely messes with everything else once you get those two involved.
Yes, exactly.
Okay, Scott, this has been really fun to talk about. Thanks for coming.
Yeah, it was a blast, and it was as much fun as I thought it was going to be.
Scott Merrick is an assistant professor in the Mallinckrodt Institute of Radiology at WashU School of Medicine. You can find a link to the paper we discussed and a related perspective at science.org/podcast. That concludes this edition of the Science Podcast. If you have any comments or suggestions, write to us at sciencepodcast@aaas.org. To find us on podcasting apps, search for Science Magazine. Or, as always, you can listen on our website, science.org/podcast. This show was edited by me, Sarah Crespi, and Kevin McLean. We had production help from Podigy. Our music is by Jeffrey Cook and Wen Koi Wen. On behalf of Science and its publisher, AAAS, thanks for joining us.